Welcome to the problem-solving set on gene interactions, epistasis, and modified Mendelian ratios. Question 1 states that the production of red flower color in a certain plant species follows the biosynthetic pathway below, where gene X takes white pigment to pink pigment, and gene Y takes pink pigment to red pigment. Using this information, determine the ratio of offspring phenotypes for the following crosses. So A, it is big X, big Y, times big X, big Y, little y. So this, A, is a normal dihybrid cross. So let's write out our phenotypic ratios for the offspring. With a dihybrid cross, you get 9, 3, 3, 1. Where the 9 is both genes having at least one dominant allele. 3 is where one is homozygous recessive and the other has at least one dominant. The other 3 is the opposite, so the X would be big X dash little y, little y. And then the 1 corresponds to both being homozygous recessive. So let's see what so these are the genotypes, right? Let's see what the phenotypes would be for this cross. If an individual plant has one big X and one big Y, that means they would make both of these products here. They would make both of the enzymes that take each colored pigment to the next. So that means it would produce red pigment. So let's write red. This 3 has two little x's, which mean it does not produce the enzyme X. So therefore, these plants would be white. They would have no enzyme to take the white pigment to the pink pigment. For this 3, it has one big X, which means it will make the gene X product, but it has two little Y's, which means it will be missing that product and will therefore be pink. And finally, being homozygous recessive at both loci means that they would have no gene product to take the white pigment to any other color, so they would be white. So the phenotypic ratio for this cross offspring would be 9 red to 4 white to 3 pink. For part B, both parental individuals are homozygous dominant for the X gene. So that means that all of the offspring individuals will be homozygous dominant for the X gene, and therefore there will be no white individuals in the offspring. So let's do basic probability. So the probability of having XX, big X, is 1. Then for the Y locus, you have little y times big Y, little y. And if you do the Punnett square for that, you see that half of the time you would have big Y, little y, and half of the time the offspring would have little y, little y. So if you do the line method here, you would get half of the offspring having two big X's, one big Y, one little y, and one half of the offspring having two big X's and two little y's. Now for these individuals, two big X's, you would definitely make the pink pigment, but then they would also have the gene Y product to go ahead and make the red pigment, so you would have half red offspring. For the second half of the offspring, you would have the gene product to go from white to pink, but then you're missing the gene product for gene Y, so you would stay at pink. So the phenotypic ratios for the offspring are half red to half pink. So let's just write that on top so we keep track. So it would be 1 red to 1 pink, and from before it was 9 red to 4 white to 1 pink. I'm sorry, 3 pink. For part C, both parental individuals are homozygous recessive for gene X. Now that means they don't have the enzyme that converts white pigment to pink pigment, and all of their offspring will both be homozygous recessive as well for this gene X. So all offspring will be little x, and here it doesn't matter what alleles they get for gene Y, because if they don't have the enzyme that converts white pigment to pink pigment, they can't ever produce red pigment because it's in the biosynthetic pathway that flows in that certain order. So 100% of these offspring will be white. So let's just say 100% white. Okay, so for part D, we're going to do the line method again. So let's look at gene X. You have big X little x times big X little x, which is a simple monohybrid cross. So you would have 1/4 big X, 1/2 big X, little x, and 1/4 little x, little x. Now for locus Y, it is little y times big Y, little y. So therefore you would have half big Y, little y, and half little y, little y. And you write that for every x that we had. Okay, and let's now multiply across. So you have 1/4 times 1/2. It's 1/8, where you have big X, big Y, little y. 1/8 big X, little y, little y. 1/4 big X, big Y, little y. 1/4 big X, little y, little y. 1/8 little x, big Y, little y. And 1/8 little x, little y, little y. So these are the genotypes. Let's look at the phenotypes for these offspring. Okay. Now this offspring, the first one, has one big X and one big Y, which means they'll make both gene products and get a red pigment. This offspring has a big X, but is missing a big Y, and therefore will only go so far as making a pink pigment. This individual has a big X and a big Y, and again will be red. And this individual again only has one big X and is missing a big Y, and will be pink. Then both of these individuals are missing any X and won't make the gene X product, and therefore they will both be white. So if you want to count them up, it would be 1/8 plus 1/8 be 1/4 white. Then for red, you'd have 1/8 plus 1/4, or 2/8. And that's 3/8 red. And for pink, you have 1/8 plus 2/8 is 3/8 pink. So to get these all in the same denominator, this would be 2/8 white. So you have 3 pink, 3 red, and 2 white. And that's your final ratio for that cross. Question 2. Black fur on a chocolate lab requires the dominant allele B, while the recessive allele little b produces brown fur. There is an epistatic gene, A, that inhibits pigment formation and leaves the pups albino. The recessive allele, little a, permits pigment production and inherits to fur. For the following crosses, determine the phenotypes of the parents and the resulting offspring, including ratios. So let's define our alleles first. So big B is the dominant allele, and that is for black fur. Little b is recessive, and that is for brown fur. Now the epistatic gene, big A, leaves the pups albino because the pigmentation can't adhere to their fur, whereas the recessive little a allows for pigmentation. So pigment is good. Okay, so for A, let's look at that cross. It's little b, little a, times big B, little A, little a. So the first question is, what are the parental phenotypes? So little b, that encodes for brown fur, and little a, says that yes, pigment can attach to the fur. So this parent is brown. What about the second parent? Big B, little b makes you think that it would have black fur. However, having one big A means that it is albino, which is the dominant phenotype. So this parental is albino. Now to find the phenotypes of their offspring, let's use the line method, because there's two genes interacting. So let's look at the B locus first. If you cross little b times big B, little b, you'll have 1/2 chance being big B, little b, and 1/2 chance of being little b, little b. Now for the A locus, you have little a times big A, little a. So that's 1/2 chance big A, little a, and 1/2 little a, and the same for down here. Now, multiplying these across, you get a 1/4 chance being big B, big A, little a. 1/4 big B, little a, little a. 1/4 little b, big A, little a. And 1/4 little b, little a, little a. So these are the genotypes. Now what are the phenotypes? For this one, you have a big B and a little b, which makes you think black fur. However, there is one big A, which means these pups are albino. Down here, a big B, little b is for black fur, and little a means yes, the pigmentation can occur. So this would be black furred offspring. Down here, little b is for brown, but one big A means they're albino. And little b is for brown, and little a is for pigmentation, so these would be brown. So the ratios of the offspring for cross A would be 1 to albino, to 1 brown, to 1 black. So for cross B, you have little b, little a, times big B, little a, little a. So what are the phenotypes of this parent? Little b is for brown fur, and little a means that pigmentation can occur. So this parent has brown fur. A big B, little b is for black fur, and little a means pigmentation can occur. So this is black fur. Again, let's do the line method. For locus B, there's a 1/2 chance of the offspring being big B, little b, and a 1/2 of being little b, little b. Now for locus A, both parents are homozygous recessive, which means they can both be pigmented. So for each, there is a 1 chance, 1 probability of being little a, little a. So when we multiply that across, you get 1/2 big B, little a, little a, and 1/2 little b, little a, little a. So what are these phenotypes? Both can be pigmented, so let's just look at the color for gene. So this one, big B, little b, would mean black, and little b would mean brown. So the phenotypic ratio of these offspring would be 1 black to 1 brown, and that just means they are equal. Okay, now finally for cross C, we have little b, big A, little a, times big B, big A, little a. What are the phenotypes? Again, little b is for brown, but they have 1 dominant allele for the albino gene, and therefore they're albino. And that's the same for the other parent as well. Even though they have 2 big B alleles, which would encode for black fur, they have 1 dominant allele at the albino gene. So those are their phenotypes. Now let's do the genotypes of the offspring. Big B times little b is a 100% chance of having offspring be heterozygous. So let's write the probability of 1 for big B, little b. Now for the A locus, it's big A, little a, times big A, little a, which is a monohybrid cross for that locus. So you have 1/4 big A, homozygous dominant. 1/2 big A, little a, heterozygous. And 1/4 little a, which is homozygous recessive. So then if you multiply that across, because the probability for the B locus is just 1, they're all going to have the same alleles at the B locus. You have 1/4 big B, big A, 1/2 big B, big A, little a, and 1/4 big B, little a. Now what are these phenotypes? Here, they have 2 dominant alleles at the A locus, which means albino. The heterozygotes have 1 dominant allele at the albino locus, which again means albino. But here, they're homozygous recessive at the A locus, which means you can have pigmentation. And a big B means that they'll have black fur. So for this cross, you get 3 albino to 1 black.
Hey guys, it's Professor Schmidt. Question 3 reads, feather color in a particular species of bird is determined by 2 incompletely dominant alleles. The allele FB produces blue feathers, the allele FG produces gray feathers, and the heterozygotes have rhythm, in other words, blue-gray feathers. I don't know what the rhythm part means, quite frankly. 2 other epistatic genes, O and C, are both required for any coloration in the feathers, any. Homozygous recessiveness at either locus results in white-feathered birds. For the following crosses, determine the genotypes of the parents and their offspring. So let's pause for a moment, because sometimes I tell you guys I like to not really look at the questions too much before I try to solve them, so that I can sort of go through a thought process with you about how to do so. Well, I did that on this one, and I got totally screwed up, so I had to go think about it a little bit more. And I'm not ashamed to admit that, because these things are complicated sometimes. So, you know, I guess the lesson is that sometimes you can go wrong, and you could spend a lot of time going wrong. But don't get defeated and discouraged, because if you do what I did and just go and think about it for a minute, chances are you'll figure it out. If not, you can watch these videos to give you some help. Well, let's see what we do know. And there are a couple of things in the question that I think really should stand out, right? First of all, we see we're dealing with incompletely dominant alleles. So let's just write down what we know about those. They told us, right, that FB, and you guys know that there are several ways you could write in theory incomplete dominance, but this is just as good as a way as any. They're just sort of different versions of the F gene, but without any particular one is not dominant completely over the other, right? But think of it in terms of genotypes. There's really only 3 that there could be, right? So what they're telling us is, by saying that the FB produces blue feathers, the only way to get a blue feathered bird would be for it to be a homozygote, right? FB. And we know at the other end of the spectrum, we have FG. They say produces gray feathers, but the only way to actually have that is to have a homozygote. And why am I saying that? Because we know that with incomplete dominance, the heterozygote is going to be somewhere in between the 2, right? So they call this rhythm. I honestly don't understand that, but it's blue-gray. It's somewhere in between the 2 extremes of the homozygotes, right? We can write rhythm, but if you keep in your mind blue-gray, I think you'll have more of the feel for the fact that this is incomplete dominance, all right? So if that's all it was about, it would not be bad at all. But the other part, and this is, you know, it really is a good question, albeit potentially frustrating, is this part, 2 other epistatic genes, O and C, and here's the very important thing. They're both required for any coloration in the feathers. And they even tell you exactly what they mean by that, that homozygous recessiveness at either locus gives you white. That's it. So forget about, keep this in the one side of your mind. I'm not sure if you've watched everything so far, but I mentioned in one of the theory videos that you can think of, in this case there are two genes, but here's the actual color that's going to be made, right? And in this case the other two genes, really what they're saying is turn the switch on or turn the switch off. If the switch is on, then these colors are going to be produced by those genes. If the switch is off, it's pretty much irrelevant what's going on over there because you're going to have white, right? Now, so we have to ask ourselves what's necessary for the switch to be on and what's necessary for it to be off. They say homozygous recessiveness at either locus results in white feathered birds. So in other words, if you have, say, big O, it doesn't matter what the other one is, but if you have little c, that switch is off. You're going to have white, right? The same type of thing. If you have little o, and a big C and anything else, it's also going to be white. I mean, even if you have all homozygous recessive, it's still going to be white. So that tells you what you need to have it on is at least one big O and one big C, right? We're assuming the working dominant versions of those genes are both necessary somehow to turn the switch on. And once it's on, as we said, then we can consult over here to see what's going on. All right? Armed with that knowledge, and you can think of them sort of separately in terms of the crosses. So let's look at A. A is not so bad. B is the one that's bad. Two white birds produce blue-feathered offspring. So you have to think, how could this happen? If they're white, there could be a lot of reasons that they're white, which we'll talk about in a minute. But if they're producing blue-feathered offspring, we know something about what the offspring have to be. And just for the record, we need to, what they're really asking us is based on the data to tell what the genotypes of the parents and the offspring are, right? So I'm going to switch colors here and go down a tiny bit. Okay. Because I want to be able to say, let's think about the offspring in this case. What does a blue-feathered bird, even though I'm writing it in red, what does a blue-feathered bird have to be like genetically? Well, we said if it's blue, it has to be like that, right? F big B, F big B. So we know the offspring have to be that way. Now, if the color switch is on, and it has to be if you're getting blue-feathered birds, that means we just said we have to have at least one big O and one big C, right? So the question then becomes, if the parents both were white, how could that be? And how could they reconstitute, if you will? In other words, in the two parents, basically what they're saying is the switch was off. But in the offspring, the switch was on, right? Now, if all the offspring were blue, I hope you guys, this is the F1, right? The offspring. Now let's think about the parents, going backwards a little bit here. The parents were white, but does it not have to be true that both of the parents at this locus had to be FB, FB, right? If they had anything else in there, then in the offspring, you would probably be finding some of the gray and some of the rhythm or something like that. So in essence, if all the offspring are blue-feathered, you need to assume that the parents both potentially were blue-feathered. It's almost like a pure breeding line, except for the fact that something was up with the other genes, the O and the C, the switch genes. Okay? So you have to envision yourself, how could I have a situation where the switch is off in two different birds, but when you cross them, the switch comes back on? And think in your mind where else are you going to do it. One way that can happen is, and we've seen it before, is that, look, think of it this way. The offspring have to get a big O from someplace, right? So one of the parents has to have a big O. Let's say it's this one. If it has a big O, why is the switch off? Well, it's going to have little c, little c, right? As we saw over here, that would be an example like this. Now, we don't really know what the other one is, but we're going to be able to figure out in a minute. Let's just continue with this logic. So this is, one parent here. This offspring has to get a big C from someplace too, right? So it's getting it from here. But then why would those birds be white? They have to be little o, little o. Now, if you really think of it, what you're doing is a dihybrid cross, and if this was, it turns out that what this has to be is big O over here, and big C over here. And if you think about the reason is, you're doing a dihybrid cross, and if there was any of the small letter here, there'd be a chance for some of the offspring to be white. So let's think about this. So if this is true of the parents, we can do the crosses separately. This one we don't even have to do. I'll switch colors again. What I mean is, if you cross FB times FB, you're obviously getting FB, right? So those are the genotypes of the parents and the offspring with respect to that locus. Now, we can do this though. If you do this cross, big O, little c, times little o, big C, even without doing a Punnett square, I know you guys can see, because you've done it enough already, that the offspring will always get big O from there and little o from there, big C from there, little c from there, and that all the offspring will be big O, big C, little c. So we could fill that in over here. Remember, all that you need for the switch to be on and the blue color to manifest itself is at least one big O and at least one big C. So guys, really this is the only way it could possibly work, and that is the answer. I don't want to ruin it, but just think about this, because what I said before, some of you might be thinking, well, how did you jump to the conclusion all of a sudden that this had to be, we knew this had a big O and two little c's, but what if it had a little o, right? Well, you can see that half the time here, this little o would get together with this little o, and you would actually end up having homozygosity of the recessive at that locus, and that would be a white bird. But remember, all the offspring are blue feathered here. Okay? Rather than extend the page, I would prefer to erase all this. So I'll leave it for 1 second. Of course, you could always pause or rewind. If you want to look at it for a while, pause now. Okay. So I'm going to erase all of this. This that I'm leaving for a second, that's always still going to be true. In fact, might not hurt to leave that one for a second, because that's always true, right? Just showing us what kind of genotypes. If there are colors being produced, what's going on here at the incompletely dominant locus. So for B, 2 blue birds produce 93 blues and 68 whites. Almost the opposite of before. The parents are blue, both of them, and the offspring have some blue and some white. I'm going to admit to you guys that this is the one that sort of messed me up when I first looked at it, but here is the key. Well, first let's look at what we know about the genotypes of the parents, and possibly what we know about the genotypes of the offspring. 2 blue birds. So if they're blue, we know, sort of just like we saw before, that's parent 1. All right, parent 2 over here. To be blue, you have to be FB, right? So we know what was going on with the 2 parents at that locus. And in fact, doesn't that mean that all of the offspring, the F1, are also going to be F big B? So it is sort of like the last cross in a sense. But now, the reason why it's somewhat opposite and a little bit strange also is that the 2 blue birds, so we know the color switch was on in the parents, right? And what that means is both the parents had at least one big O and one big C, right? What about the offspring? Well, if it's a blue bird, the color switch is on sometimes. If it's white, then we know the color switch is off sometimes. So that tells us, listen, The only way for the color switch to be off is if you have, just as an example, big O over here, right? Doesn't that mean that all the offspring, regardless of what's going on over here, they're all going to have at least one big O, right? And doesn't that mean the color switch is going to be on? And the same logic goes for the C. So it's really necessary to postulate that these parents are double heterozygotes, right? You see why? If they were homozygous for the dominant at either spot, then all of the offspring would have the color switch on and all of them would be blue. But some of them are, but some of them are white too. Now, it turns out that the numbers here are extremely important. 93 to 68. And what kind of ratio is 93 to 68? Well, it may not be immediately obvious, and this is the part where I got stumped initially. If you do the math, it's not exact, but clearly this isn't a 3 to 1 ratio, right? Because 3 to 1 would be, quickly, 34, 68. It would be like 102, something like that. If this was 68, that would be 102. Now, that's not out of the question completely, but it turns out that this is a 9 to 7 ratio, and that's really the key to the whole thing. If you watch the theory video, when you have a 9 to 7 ratio, it was the same type of thing. You needed at least one, well, we didn't use O and C necessarily, but you need one big O, one big C. And if you're doing this cross, big O, big C times big O, big C, little c, remember that's a dihybrid cross. And if it's a double heterozygote as it is, you're always going to see something in the nature of a 9 to 3 to 3 to 1 ratio. And remember that a 9 to 7 ratio is combining the 3, and the 1. So the 9 are going to be those that are like that. Just so you remember, these 3 are going to be like that. Those 3 are going to be like that. And this little one is going to be like that. This is exactly what we're seeing here. We're seeing that if the parents are like we have said that they almost definitely must be based on the data, we should get a 9 to 3 to 3 to 1 ratio. But remember, you need both these genes to turn the color switch on. They're acting together. So it makes sense that in a 9/16 of the offspring, you have one big O and one big C, the color switch is going to be on. In all the rest, you have what the question told us, homozygosity recessive at least one of the loci, and maybe two. Those are all going to be white, right? So what that means is this isn't that bad once you come to that realization, because the bluebirds, we know they have to have the color switch on, right? What I mean is these 93 blue offspring. So we wouldn't even need to do any cross to say that they're big O and big C. Now, here's the thing. So the ones that are blue are going to have at least one big O and one big C. Put on hold for a minute what the exact genotypes of those birds might be, because I want to focus on the white birds. The white birds are, there are a lot of ways that they could be what they are, but they're still FB, right? Just that the switch is going to be off. So any of these three categories will work, meaning it could be little o with a big C. It could be, little c with a big O. And it could be complete homozygosity there. So at this point, I guess the reason I hesitated just for a moment is this is usually enough of an answer for anybody, okay? In other words, to say the blue birds have at least one big O and one big C, whereas the white birds, sorry, I made a little mess up over here. The white birds are like this, because the whole point of this problem is to get you to see that it is a 9 to 7 ratio of those on-off switch genes. If you really needed to know the exact genotypic ratio of every single one there, you'd have to do a Punnett square, and you'd have to see the 16 boxes, and you'd have to give the ratios of all of the different types that can have one big O and one big C. There are a bunch of different ones. I don't know if it's that profitable for us to do that right now, because this is really the key of what the problem was getting at. Doing that would be some work, but it's not terribly difficult work. Actually, I won't do it, but I'll just show you how to set it up in case you forgot. Basically, we're doing this cross, right? And when you make the Punnett square, remember, there are going to be able to be 4 possibilities with respect to the gametes on both sides, since both parents are the same. And then I didn't make it really wide enough, but you'd have to fill in all those boxes and then find 9 of them are going to have at least one big O and one big C, but there's going to be certain genotypes. Like this one is all big O, big C, right? This one over here, just as an example, would be big O, big C, little c. So if you really want to take it that far, or if you're told to take it that far, that's really what you'd have to do. There is a way to do it with the line method as well, but this is sort of going back. See, in this chapter, we're really all about the epistasis, the incomplete dominance, stuff like that. So this is going back to a basic sort of dihybrid cross situation, and I just don't think we need to spend any more time on that here. All right, so this is the key, is recognizing that 9:7 ratio and realizing that, you know, it is in fact a dihybrid cross. So again, if you need to look at that a little more, well, you can pause now, because I'm going to erase it. Okay. You guys gotta admit, this is fun. I don't know what else you could ask for from life. Well, it's fun sometimes, to be balanced with other activities. All right. Lastly, as if this weren't all enough, I'm also going to erase, I was circling some things in the questions. Let's go be green and go to C, and see that 2 rhythm birds produce 14 blue, 43 rhythm, and 16 gray. Even though there are more categories here, I hope you'll see right off the bat that this is an easier one, because is it not true that in all of these, if they're colored, the color switch must be on? And the parents being rhythm birds, the color switch must be on in all of them. So that means, forget about this for a second, all of them have to be actually homozygous for the dominant at both locations. This is the cross we're doing. And the reason it has to be that way, if there was a little O, well, genetics is hard. Something always has to make an easy rule not necessarily quite so easy. It is true that if one of the parents was like that, right, all of the offspring would at least have one big O. And if one of the parents was like that, all of the offspring would have at least one big C. So if you really wanted to get very specific about it, you could postulate that there are some possibilities. But if you postulate that both parents are like this, then we know for a fact that if you want to think of it this way, that these birds are pure breeding not for a trait, but they're pure breeding, if they're like this, for having the switch on. And since the parents and all the offspring have the switch on, that's a very reasonable idea to make. But you could, again, if they really wanted to ask you, what are all the possible genotypes, what you'd have to do is set it up so that there's no possibility of getting any offspring that have, for example, little o. So you couldn't have 2 parents that were like this, because 1/4 of the time those 2 O's would go together. So I think it's safe to say that you'd have to have at least one of the parents being big O, big O. Now, it could be the same parent or it could be the other one. That's big C, big C, and then it's possible that you could have this situation as well. And it could be in one parent was totally homozygous, it could be the other one was like this. That's really the only other situation I can envision, but this is certainly the easiest and the best answer. Bottom line though is the color switch is on in all of them, so we don't really have to worry about it that terribly. This really is more a question about the 14, 43, and 16, right? So it's really, I mean, I hate to say it's really easy because you know what the genotypes of the parents are with respect to this locus, their rhythm, right? So the cross is this one. And we can very simply do a Punnett square. Probably don't even need to do the Punnett square, but FB, FG, right? FB, FG. So we know from this that approximately 1/4 the offspring are going to be blue. Approximately 1/2 the offspring are going to be rhythm, right? These two boxes here. And then another 1/4 approximately are going to be gray. So we have predicting a 1 to 2 to 1 ratio of blue, rhythm to gray. Now if you really look at the numbers, they're a little more rhythm than looks like 1 to 2 to 1. But they gave you the cross, you guys. So you know what it has to be. 2 rhythm birds give you 1 to 2 to 1 ratio. I mean, you guys know in real life that the actual numbers don't have to be identical to a 1 to 2 to 1, but there's nothing else that this could possibly be. So you really, in all of these, you're doing 2 crosses. You're doing the one that has to do with this locus. You're doing the one that has to do with these loci as well. And in this case, 2 rhythm birds, they told us exactly how the incomplete dominance works, so there's no other choice in this case, okay? Well, I hope you enjoyed that as much as I did, and I'll see you again real soon.
Question 4. Chicken comb shapes are determined by 2 genes that can be involved in non-allelic gene interaction. The dominant allele of one gene, R, determines rose comb shape, while the dominant allele of the other gene, P, produces a pea comb shape. A walnut comb shape is observed when at least one dominant allele for both genes is present. When both genes are homozygous recessive, a single comb shape is observed. A pure-breeding pea comb chicken is mated with a pure-breeding rose comb rooster. What are the genotypes and phenotypes of their offspring? So first, let's look at the genotypes of these parents. So you have a pea comb chicken. Now again, in order to be pea comb, you need at least one big P, and the R has to be homozygous recessive. So this is little r, big P, big P. And we know it's big P because they're pure breeding. So if you cross these with themselves, you'll always get pea comb shaped. Now for the rooster, they are rose combed. So to be rose combed and pure breeding, you have to be big R, little p, little p. So if you cross these parents, all the offspring are going to be big R, big P, little p. So that's the genotypes. Now what is this phenotype? They have one big R and one big P. And when you have both genes having one dominant allele, you get a walnut comb shape. So this is walnut. So that's the answer for A. So they're all 100% big R, big P, little p, and have a walnut shaped comb. Okay, now it says for B. If the offspring from part A are crossed, what are the phenotypes of their offspring, or the F2 generation? So what we're doing is big R, big P, little p, times itself. And this is a simple dihybrid cross because both individuals are heterozygous at both genes. So when you do a dihybrid cross, you get the 9 to 3 to 3 to 1 offspring ratio. So this is the F2. Now again, the 9 represents having one dominant allele for both genes. The 3 is one dominant allele for one gene and homozygous recessive for the other. The other 3 is the opposite. and then the 1 represents homozygous recessive for both genes. So these are the genotypes, but they're asking in the problem for the phenotypes of the offspring. So let's see. If they have at least one big R and one big P, that means that they have a walnut comb shape. If they have one big R and two little p's, that is a rose comb shape. If they have two little r's and one big P, that is a pea comb shape. And if they're homozygous recessive at both genes, it is called a single comb shape. So this is the answer for part B. Scroll down part C. It says a cross between a rose-combed chicken and a walnut-combed rooster yields 15 walnut-combed, 14 rose-combed, 6 pea-combed, and 5 single-combed chickens. What were the genotypes of the parental individuals? So let's see what we know. We know some of their genotypes. So the parents are rose-combed and walnut-combed. Now we know to be rose, you need at least one big R, and you have to be homozygous recessive at the P gene. For walnut, you have to have one big R and one big P. So let's see what their offsprings were. So this is the parental generation. Now for F1, you see 15 walnut combed, 14 rose, 6 P, and 5 single. Now these are the phenotypes, so what are the possible genotypes for these offspring? Again, for walnut, you have to have at least one dominant allele at both genes. For rose, you have to have at least one big R and two little p's. For P, you have to have two little r's and at least one big P allele. And for single, you have to be little r, little p, little p. So how could we fill in the dashes for the genotypes of the parents that could give these offspring? Well, we know, let's look at the single comb, because this being homozygous recessive at both genes can tell us a lot. So we know that one of these little p's can come from the rose parent. But the other little p must come from the other parent, which means that this dash right here must be a little p. Now to have little r, we know already that both parents have at least one big R. But in order to get these little r's from both parents, that means that both of these dashes must be little r. So now we know the genotypes of the parents. The parents, again, this is the rose, big R, little p, little p. And the walnut was big R, big P, little p. And if you want to do a simple line method way to just double check that the data is consistent with these parents, let's do that. So it's big R times big R, little r. So for the R gene, you have, it's 1/4 big R, 1/2 big R, and 1/4 little r. Now for the P gene, it's little p times big P, little p. So it's a half chance of big P, little p, and a half chance of little p. And you just write that for all of the r's that we had. Okay, now we're going to multiply across. So 1/4 times 1/2 is 1/8 big R, big P, little p. Another 1/8 big R, little p. Then 1/2 times 1/2 is 1/4 big R, big P, little p. And 1/4 big R, little p. And now for this one, it's 1/8 little r, big P, little p. And 1/8 little r, little p, little p. So what are these phenotypes? If you have one big R and one big P, that's walnut. Two big R's and two little p's, that's rose. Again, one big R, one big P is walnut. One big R and two little p's is rose. And then little r, big P. That's P combed. And little r, little p, that's single. So let's add them all up. For the walnuts, you have you have 1/8 plus 1/4, which is really 2/8. So that's 3/8. For rose, you have the same. 1/8 plus 1/4, or 2/8, is 3/8. For P, you have 1/8. And for single, you have 1/8. So this ratio, it's a 3 to 3 to 1 to 1 ratio. And if you look back at the data, you had, let's see, 15 walnut and 14 rose. And then 6 P and 5 single. So although these are a little different, it's basically around 15, and these are around 5. And that makes sense because if you divide both by 5, you get the 3 to 1, which matches there. So the data matches is consistent with the genotypes we found, so therefore the genotypes of the parents are definitely big R, big little p, and big R, big P, little p. Question 5. A plant species needs at least one dominant allele in both gene X and Y to produce progeny with blue flowers. A plant homozygous recessive at either gene will have white flowers. A third gene, gene Z, is epistatic to genes X and Y. If the plant is homozygous recessive for Z, then the flowers will be yellow regardless of the alleles at genes X and Y. The following cross is done. What are the phenotypes of these individuals? So this individual is big X, little y, big Z, little z. So let's look at what we would guess if we were just looking at X and Y. So let's highlight the important information here. So you need at least one dominant allele in both gene X and Y to produce blue flowers. If you're homozygous recessive at either gene, you will have white flowers. Now for gene Z, if you're homozygous recessive for Z, then the flowers will be yellow. Okay? So let's look at big X, little y, little y. So it's saying if you're homozygous recessive at either X or Y, you will have white flowers. So just looking at these two genes, you would assume white. Now let's look at the Z gene. It says in order to have yellow plant, you need to be homozygous recessive at Z, and this is heterozygous, so therefore you wouldn't be yellow, and this parent would be white. Now the other parent is big X, big Y, little z, little z. So again, let's first look at X and Y genes. So they have one dominant allele for both X and Y, so you would guess blue flowers. However, at the Z gene, they are homozygous recessive, which means that they're yellow regardless of what's at X or Y. So these parents are yellow. Okay. So for A, white and yellow. Now for B, what is the ratio of phenotypes in the F1 generation? So let's do the line method. I know that gives us all the genotypes, but because it's, a trihybrid cross, you definitely don't want to do the Punnett square. So let's do the simple line method, and then we'll fill in the phenotypes after. So let's look at the X gene first. You have big X times big X, little x, which is the monohybrid cross. So you have 1/4 big X, 1/2 big X, and 1/4 little x. Now for Y, you have little y times big Y, little y. So there you have a half chance, big Y, little y, 1/2 little y. Same here, half. and the same down here. Okay. Now for Z, you have big Z times little z, little z. So again, it's going to be 1/2 big Z, little z, 1/2 little z, little z. And that's the same all the way down, so just go out, fill that out. Sorry, my z's are looking like 2's. I'm sorry. Should be little z, little z. Okay. Now let's go and multiply through and get the genotypes. So we have 1/4 times 1/2 times 1/2. So that is 1/16. Big X, big Y, big Z, little z. Now again, you're just going to follow straight across. So it's 1/4 times 1/2 times another half. Again is 1/16. Big X to one big Y. These line methods get confusing when there's 3 genes. Okay, big Y, and now little z, little z. Now these are little y, little y. Okay. Now we have 1/2 times 1/2 times 1/2. So that's 1/8. Big X, big Y, big Z, little z. 1/8. Big X, big Y, little z, little z. Now we have little y, big Z, little z. Okay, and then finally the last row. Now it's 1/4 times 1/2 times 1/2, so it's 1/16 again. And you have little x, big Y, big Z, little z. 1/16 little x, big Y, little z, little z. 1/16 little x, little y, big Z, little z. And 1/16 little x, little y, little z, little z. Okay, so these are the genotypes. I know that, sorry that took so long. But now we have to go through and look at the phenotypes for these. Okay, so again, let's just refresh our memory. You need at least one dominant allele in both X and Y to have blue flowers. If you're homozygous recessive at either X or Y, you're going to have white flowers. However, if you're homozygous recessive at gene Z, then you'll have yellow flowers regardless of what's at X or Y. So why don't we go through and look at which plants are homozygous recessive at gene Z, so we know that those are yellow and it doesn't matter what their X or Y's are. Okay? So let's see. Which ones are homozygous recessive at little z? So we have here, it's yellow. Here is yellow. It's basically every other one, seems like. Yellow. Yellow. And then this last one is yellow. Okay. Now, you need at least one dominant allele in both genes X and Y to have blue flowers. So let's look for that. One big X, one big Y. That's blue. And of course, these are the ones that aren't already yellow because of gene Z. So now we have one big X, one big Y. That's blue. And those are all the blue flowers. Now, if you're homozygous recessive at either X or Y, you're going to have white flowers. So here, homozygous recessive at Y, white. Again, homozygous recessive at Y is white. homozygous recessive at X this time, still white. Here they're homozygous recessive at both, so they're white. And that's all of it. So let's add up. So we either have yellow, white, or blue. So for the yellows, we have 1/16 plus 1/16 plus 1/8. Let me erase this. Okay, so we got these three. Then we have another 1/8. And then we have two other 1/16. So again, let's cross these out and make them 2/16, so it's just easier to add up. Okay, so we have 1 plus 1 is 2, plus 2 is 4, plus another 2 is 6, plus 1 plus 1 is 8. So this is 8 out of 16 are yellow. Now let's look at white. We have 1/16, 1/16, plus 1/8, plus 1/16. I think that's all of them. Okay, so if you add those across, you have 1 plus 1 is 2, plus 2 is 4, plus 1 is 5. So 5 out of 16 are white. And then finally we have blue. So we have 1/8 plus 1/16. And that is 3/16, or blue. So in total we have 8 yellow to 5 white to 3 blue. And as you can see, this is a modified Mendelian ratio for a dihybrid cross.